So just before we start, I have to say a couple of words. So first of all, it's a big pleasure to be here, and I have to say I'm probably the most odd person here because not only I'm not a theoretician, but rather an experimental side of things, but especially recently I'm straying further and further from my physics roots and moving more and more in biology. So most of the research which I'm personally doing is actually on the proteins. But then when I've got an invitation from Nikita on this conference, I was thinking if I start talking about the proteins, then most likely I will lose 90% of you within the first 2 minutes. So I decided to maybe have a talk on a little bit more physics side of things. So, and then I remembered that I was a couple of years ago a postdoc at the European EuXFEL and decided to make a presentation on the method. So just a couple of voluntary disclosures. So now I'm a postdoc at the European Molecular Biology Laboratory. So I'm much more of a biologist nowadays. So when I present to the biology people, then they usually say that I'm a biophysicist. And if I don't know something about the biology, then I'm a physicist. That's why I don't know. So here I want to say that, of course, I'm much more of a biologist now. So if I don't know something about the physics, then you should blame it on my new biology background, apparently. So, okay, so what I will be talking about today is the theory of femtosecond crystallography. And later we will touch the EuXFEL side of it, because of course, if we say the word femtosecond, especially in an experimental context, then you need to touch EuXFELs. We'll discuss what are the cases of theory of femtosecond crystallography in biology, why do you need it, how to approach it, and what is the point of doing these experiments. So when we start talk about the theory of femtosecond crystallography, we need to approach it block by block. What's serial, what's femtosecond, and what's crystallography? So very first question, of course, is what is crystallography? So when we talk about the X-ray crystallography, you need a couple of things. So if you come to the synchrotron or EuXFEL, you need something to measure. So you need something which is crystalline, because crystallography, by definition, needs some crystals. You need some periodic structure on which you shoot your X-rays, ideally. I mean, generally speaking, light, but in this case, of course, X-rays. And that what you aim for is you want to receive some form of these diffraction peaks or Bragg peaks on your detector. And usually when you talk to people with a physics background, especially in this X-ray field, if you ask them about the presentations, they start from the Maxwell equations. But here I will start talking from the Bragg equation, of course. So here what we actually approach when we start to numerically solve the diffraction patterns is that this diffraction image can be converted to the electron densities by using the form of the Fourier transform. And this was actually, let's say, numerically a pretty hot topic maybe 20 years ago, but nowadays, especially with the advent of the molecular simulations and more importantly, the ML and AI. So there was a program which was called AlphaFold, which managed to provide us with more or less reliable structures for each protein. So now there is no such thing as a phase problem, which means that if you collect the diffraction for your protein, you'll be almost certainly able to solve it and get your structure. So nowadays, people are moving much more towards the not just solving the structure, but trying to understand the dynamics, trying to tackle specific cases, let's say. So next, we have to cover what's the serial part of the crystallography. So when we talk about the serial part of crystallography, we have to touch a little bit the concept of EuXFEL, which I'll discuss more in a further slide. But the main point is that if you tackle the standard macromolecular crystallography, then you are measuring one big single crystal. So you have one big single crystal, you try to freeze it or do it at the room temperature, so they don't want it to dehydrate too quickly. Then you shoot a light, you rotate the crystal, and you collect the full rotational space, so you can reconstruct the 3D image of the diffraction. By using that, you can reconstruct the 3D structure of the protein. And when you talk about the serial concept, then you want to use many, many crystals. So usually you need smaller crystals. You don't need large, one big crystals. You need many small crystals. And then what you do is that you constantly move these crystals and illuminate them with your laser or synchrotron light. And then you collect many, many snapshots. And in this case, the conformational space is covered by the statistical metrics. So you're trying to collect as many snapshots as they are, and you get the full conformational space because you simply sampled it enough times. And here also it's important to talk a little bit about the pump probe concept. So the point of the pump probe concept is that you can always pre-activate your crystals with something. And typically, especially for the EuXFELs, because the only thing which can compete with light is light. So usually you couple it with a pump laser, which will pre-activate your protein, or generally speaking, your system, and then you collect the diffraction of these activated states, and then you'll try to decipher it and study it. So now let's talk a little bit more about the EuXFELs. So when we talk about the EuXFELs, so EuXFELs is I mean, now there are multiple people which know the topic probably even better than me, especially from the theory side. But if you talk more about the practical side, then we have synchrotrons, which are much more cost efficient in terms of the experiment, because you can build your ring and then put many beam lines on it. So typical synchrotrons, they have around 30 beam lines, sometimes more. But for the EuXFEL, it's actually not the case, because you're always competing for the same light generated by your source. So in that case, each EuXFEL can only facilitate. So for instance, on the European EuXFEL, there are only 6 instruments, and typically there are, let's say, around 5-ish instruments. In case of SwissFEL, for instance, they only have 2 branches, so only 2 instruments, but a couple of end stations there. And what's the advantage of the EuXFELs? So I'll not go too much into details explaining how exactly it happens, but the EuXFELs are capable of generating a much higher brilliance and much shorter pulse. So what you get in comparison to the synchrotron sources, you get much more intense pulses, much more bright pulses, which are also much more localized in time. So when you're talking about the synchrotrons, so the typical time with the, let's say, sufficient pulse energy would be about hundreds of microseconds. When we're talking about EuXFELs, it could be as short as the 20 femtosecond with a reasonable pulse. And then there was the discussion, of course, earlier that it can go up to attoseconds. But then, of course, you lose quite some of your intensity, which is critical for the biological applications. But for material science, you can do attoseconds. I mean, I learned it yesterday. And also, there is a description between the soft X-rays and hard X-rays, and in case of biology, of course, in hard X-rays. So there are only a few EuXFEL facilities in the world, which makes them even more unique. And of course, one of them is the European EuXFEL in Hamburg, which is very close to us. But technically speaking, there are multiple more. So there is a soft X-ray facility, FLASH, or CFEL, which is also in DESY. There is a SwissFEL, and also a couple in US and in Japan and China. In China, they also, I think, now have also hard X-ray, but this is a bit of an outdated map. So, and also what's important to mention, but we will not stay too long on that, is that the case of the European EuXFEL is particularly interesting because they're based on a superconducting accelerator. So now CLS2, I believe, is also based on a superconductor accelerator, but for quite a while, they were based on a non-superconducting accelerator, and they were generating pulses with a 100 hertz efficiency, but there was only one pulse. So what European EuXFEL is doing differently is that they have this train system. So there are many pulses which come in a train, which are condensed within a certain window. So typically 600 microseconds, and they come every 10 hertz. So every 100 milliseconds, which is actually quite rare. But then, of course, you get many of these pulses, so you can study something, which have additional strains, which we'll discuss later on the biological samples. So what are the applications of the EuXFEL in biology? So typically, if you try to apply for the beam time at EuXFELs, you get asked, okay, but did you do your work in the synchrotron before? Because you have to do preliminary work in order to apply to the EuXFEL. And then, of course, there are certain cases when you can really leverage the EuXFELs. And in some cases that you'll be said something like, okay, just go to the synchrotron. There is no use for your particular case at the EuXFELs. So I'll highlight a couple of very important examples. So first of all, since EuXFEL, they'll immediately destroy your sample. There's a concept of diffraction before destruction, which means that you avoid the radiation damage. This one should be taken with a little bit of an asterisk, but there will be an example on that. But generally speaking, especially for biological system, it's quite true. Second, you are uniquely suited to the small crystal. So if your protein doesn't produce big crystal for some reason, then EuXFEL is a good solution. And third is probably most important case is that you can study very fast reactions, which are not accessible at the synchrotron. So if your protein, especially the light sensitive proteins, which have a reaction on the femtosecond, picosecond, nanosecond scale, you have to go to EuXFEL. So I just want to mention so that one very important topic is that SFX fueled major project in GPCR research. So not to talk too long about it, but GPCRs are major targets. There was actually a couple of Nobel Prizes won on that particular topic, and they are very important drug targets, especially in humans. So the problem with the GPCRs was that these proteins are intrinsically not so stable. So those are integral membrane proteins, and people were not able to grow sufficiently large crystals to study them with the synchrotrons. But then with the advent of EuXFEL, especially in OCLS in US, they were able to leverage that. And there were multiple very important structures which were targets for the many drugs, and people got quite some recognition for that. There were many important papers. So next I want to talk about one topic where I'm personally was quite involved. So it's not really my publication, but we were studying this protein also in a group where I was doing my PhD. So that's the topic of the bacteriorhodopsin. So bacteriorhodopsin is also integral membrane protein. It has a light sensitive cofactor. In this case, it's a retinol cofactor, which is vitamin A. Essentially, everyone has them in their eyes. And in this particular case, it's used in bacteria for the proton transfer. So the interesting story is that this particular protein, it has a very fast initial photoreaction. So the initial photoreaction is the cis to trans, all trans to 13 cis transition. So essentially the flip around one of these chains. And the people were interested how fast it actually happens. So there were quite some, let's say, simulation, also spectroscopic studies. But then later people were able to actually measure the direct evidence of how fast it happens. And there was actually a discussion yesterday during the talk, and I promised to show something. So in this case, of course, indeed, it's not so easy to assign a proper time tag, but in this case, let's say up to 150 femtosecond, you can see that there is a significant shift in electron density. So in this case, these meshes represent the negative and positive electron density. So there's also different electron maps, which means that this density, it moved from here to here. So then you see that there was some significant transition. So in this particular case, I think it was around 0.5 angstrom. And then later it goes on and then completes the transition within essentially picoseconds. And then when they measured the speed of that, they found that it was faster than the speed of sound and water, which is somewhat not surprising for the people as I understood, but yeah, I think especially for biologists, it was quite remarkable finding. So next topic is also I find personally extremely interesting. So this is the topic of the drugs, which can be artificially converted to be photoswitchable. So the story here is that you have naturally occurring drug, which is called campristotine A4. So this particular drug is interesting for as I understood, also anti-cancer applications, but I'll not dwell too much on it, not to, let's say, make my fool of myself. But the point is that it has double bond between these two benzyl groups. And what you can do is that you convert this bond into the benzene bond, so double nitrogens. Then they start to react to light in a way that if you shoot it with, let's say, light at the different wavelengths, you can either force cis to trans transition or trans to cis transition. And that's actually extremely important for the target delivery because you can initially deliver the inactive form of this drug to the target cells and then activate it later. And then again, deactivate it whenever you see fit. And the idea here was that what you can do is that you can study how it actually works in the proteins. So in this case, they use it in a tubulin protein. So tubulin is the major protein which forms nanotubes in cells. And what they were studying is the reaction of this photoactivation. So they had a protein. It had this drug bound to the protein. And then after illumination, you can see how quickly the reaction can occur. and that it actually occurs. So by using this data, you can also optimize these drugs, and that's a very prospective line of the research for the switchable drugs. And I think we'll see in the future many more studies like that. So I find it personally quite remarkable example. Another example, which I also find quite interesting, but from the perspective of the metalloproteins, it's also a little bit from the earlier talk, let's say. And here people are studying, so this is one of the cytochromes. And here they're studying the reaction of how the cytochromes can convert nitric acid, NO, to the nitrous acid, N2O. And the main idea was that if you study it in the synchrotron, then you'll get a different result in comparison to what you will see at the EuXFEL. So there was a specific water molecule, which later you can predict there is it attaches the NO. And then what you see here is that even if you do the serial synchrotron approach, so you deliver a very, very small dose at your protein, this dose, even by being very small, is still sufficient to, let's say, shift electronic density enough so you don't recognize this true mechanism. So I put true in quotes. But the point is that even if you are very quick with your synchrotron, the method is intrinsically unable to recognize the proper kinetics here. So you really have to go to EuXFEL to fully avoid radiation damage, or at least avoid it on this femtosecond scale, in order to be able to fully recognize the conformations of the reaction. So in some cases, radiation damage can actually be detrimental for the mechanism. And in this case, the synchrotrons are just incapable of capturing it correctly. So now let's talk a tiny bit about the European EuXFEL, also partially because I was working there. So with the European EuXFEL, the story is that because of the train structure, they have to deliver crystals extremely fast. So instead of, let's say, more slower jets, they use very fast liquid jets, which essentially shoot the crystals at very high speed. And then they are captured at this very high repetition rate. So you collect data at a crazy speed, but you also spend crazy amount of sample. And there are a couple of very interesting applications from my side. There was one relatively famous, at least at the European EuXFEL experiment, when the people came from the crystals from bacterial insecticides. And the idea was that these structures were not able, at least at the time, to be properly modeled with the AlphaFold, which is the prediction module. So people were interested in the true structures, and they came with many crystals, and within essentially one day, they were able to generate such amount of data to solve really multiple structures, which is showed the capacity and capabilities of European EuXFEL as with this liquid jet approach. So if your crystals can jet at the very high speed, then European EuXFEL can really leverage the data generation in that case. And then another example, which is I find also interesting, it's somehow goes back to this bacteriodopsin story, is that there was a protein, in this case with the PCA chromophores called PYP, photoactive yellow protein. And it has a very fast photoreaction when you have a rotation around one of the bonds. And they were able to see that. In this case, it's not really femtoseconds, so it doesn't really go to this discussion from how fast they can move within femtoseconds. But they were able to really capture the kinetics and confirm the calculations that within a couple of picoseconds, the reaction was progressing extremely fast. So there was an example also of how people were studying the very fast biological reactions using the European EuXFELs, or EuXFELs in general. Okay, so I hope that this presentation probably tickled a couple of your neurons in a way that you got something to think about in that particular sense. But just to rehearse, the simple take-home messages are that serum femtosecond crystallography is the powerful method for biological applications, but only for the certain applications, not for every, let's say, biological system you can dream of. That EuXFELs generate very short and bright pulses and enable correction of radiation damage free, again with asterisk, diffraction from small crystals. That you can do experiments to confirm your, let's say, theoretical predictions at the very fast timescales, and EuXFEL is probably the only place when you can do it in terms of structural biology, so structural approach, not the, let's say, spectroscopy approach. And that these experiments are usually used as complementary experiments to already existing data, because you cannot really come blindly to EuXFEL. You have to do a bunch of experiments before coming to EuXFEL. And yeah, there is a big roadblocks because the experimental time is quite expensive also. So that brings me actually to the end of presentation. So I'll be happy to answer the questions. So this is the old photo from the group. I think now the group is actually much smaller. So I'm here, that I was actually there. And I'm happy to answer your question.